This is part 2 of 3 on chapter 4, a survey of prokaryotic cells. In the first part of this chapter, we looked at what a prokaryotic cell is, and we compared it to a eukaryotic cell. And then we focused on the structures that are external to the plasma membrane. So we talked about the different appendages, and then we talked about the different types of glycocalyx that a bacteria cell can have. And then we also covered the cellular envelope, which includes the cell wall and the cell membrane, or that plasma membrane that tells you what's inside the cell versus what's outside the cell. So in this part of the chapter, we're going to focus on the internal structures. So what structures are inside of your cell. And then we're going to also look at a few different shapes that bacteria cells can take on and how we classify different types of bacteria. So first we're going to start with looking at inside your bacteria cell. So inside of the cell, inside of your plasma membrane or cell membrane, you're going to have the cytoplasm. And I mentioned it in the first part of the chapter. The cell cytoplasm is just a dense gelatinous solution. So it's kind of like jello, watery jello. It's made up of water, so it's 70 to 80% water. And then in that water we have sugars dissolved in it, we have amino acids, and you also have salts that are all dissolved in this cytoplasm. In addition, inside of your prokaryotic cell, they're going to have what's called a nucleoid region. So remember, prokaryotic cells are really simple cells. They don't have a nucleus, but they do have a nucleoid region where you're going to find the chromosome. So bacteria cells, they have one chromosome, so it's a single chromosome. It's circular, so it doesn't have a beginning and an end. It's just a circular piece of DNA. In addition to the chromosome, bacteria cells can also have things called plasmids. These are really small circular pieces of DNA. They're not essential to bacteria growth and metabolism, but they can hold things such as genes that have resistance to certain antibiotics, or they can hold other types of genes that might help that bacteria cell survive in the environment, but they're not essential. A lot of times these plasmids are going to be used in genetic engineering, and we'll get to genetic engineering towards the end of the semester. So we're going to come back to how we use these plasmids. You can cut the plasmids, insert genes, and do that genetic engineering. Another important structure in your bacteria cell or in your prokaryotic cell is the ribosome. Ribosomes, they're made up of 60% ribosomal RNA or rRNA and 40% protein. In ribosomes, they have two subunits. There's a large subunit and a small subunit. These two subunits of your ribosome, they come together during protein synthesis. So your ribosomes are going to take your genetic information that's coded for in the DNA, and they're going to use that to make all the different proteins the cell needs. And that includes all the different enzymes and other types of proteins, like proteins that help make up the cell wall, proteins that go into the cell membrane. So ribosomes are really important for cellular function. Other types of internal structures, you can have inclusions and granules. These are just intracellular, so intracellular means that they're inside of the cell, storage bodies. And the number of inclusions or granules, it can vary in size, number, content, so you can store different things. You can store carbohydrates, so like starch, for example. You can store other types of energy sources. And the bacteria cells, they can use these storage bodies, or they use that stored energy when the environment's resources get depleted. So these allow bacteria cells to kind of survive when the environment goes bad. Another internal structure is called the cytoskeleton. This is an internal network of proteins, and they're associated with the cell wall. The cytoskeleton, it helps the bacteria cell kind of organize itself internally. And cytoskeleton, it's basically what it means. It's a skeleton inside of your cell. Another internal structure that some bacteria cells can create are called endospores, or you'll see it as spores. These are inert resting cells produced by some gram-positive bacteria, so Clostridium, Bacillus, Sporosarcana. And these endospores, we'll get to it, but endospores, they help the bacteria cell survive really, really bad environmental conditions. So most bacteria, or not most bacteria, so it's these gram-positive bacteria, the Clostridium, Bacillus, Sporosarcana. These bacteria, they have a two-phase life cycle. So when the bacteria cell is metabolically active and growing, that's called the vegetative cell. And then the other phase of the life cycle is the endospore. So that's when your vegetative cell, it's been exposed to really bad environmental conditions, so adverse environmental conditions. The vegetative cell is going to produce this endospore, and then the endospore is very highly resistant. So it's very hard to get rid of, it's very hard to kill, and it's made for very long-term survival. So examples of different endospores: anthrax spores, those are endospores. If you breathe in the anthrax endospores, they get into your lungs, they will germinate and become the vegetative cell. And the vegetative cell is what is going to make the person sick. So the formation of endospores, when your vegetative cell creates an endospore, it's called sporulation, down at the bottom of the slide. And then germination, I just use this term. This is when your endospore, it goes through germination, it returns to the vegetative state of its life cycle. And this slide is showing this two-stage life cycle. So if we start at the very top of the cycle, you have your vegetative cell, and it's numbered with a 1 in the yellow circle up at the top. So your vegetative cell, if it enters into really adverse environmental conditions, it's going to go to step 2, so we're going to go to the right, and there's a 2 in the yellow circle. So your chromosome that's in your vegetative cell, it's going to be duplicated, and it separates. So you're going to have 2 copies of your chromosome. Then if you go to step 3, your cell's separated into a sporangium and a forespore. The forespore is what is going to become your endospore at the very end. So you're just keep on going around the cycle. So step 4, your sporangium engulfs the forespore to further develop. The sporangium becomes to synthesize layers, so your spore is going to end up having 3 layers to it. And that's what helps it kind of protect that DNA from any type of chemical or dehydration or anything bad that can happen to it. So if we keep on going through the cycle, eventually you get your layers put on your forespore. So you end up with a cortex. You have outer coat layers that are built up around your forespore. And then once you have those layers, you have a mature endospore. So I'm on step number 7 right here. The vegetative cell part usually breaks apart at this point because it's no longer needed, because we have our DNA really well protected. So step 8, you have just this free endospore that's sitting around. If that endospore enters into really good environmental conditions, that's when you're going to have the germination. So your endospore goes through germination, and you end up back with your vegetative cell at the very top. So this is the life cycle of only a few bacteria. Not all bacteria produce endospores. Just a few of them produce endospores. So these endospores, they're dehydrated, they're metabolically inactive, so basically it's just preserving the DNA in this capsule. Their longevity verges on immortality. We have found endospores that we have estimated are 250 million years old. So these endospores, they help bacteria survive for a very, very long time. The other thing about endospores, they're resistant to ordinary cleaning methods. So they're resistant to a lot of chemicals we use, and they're resistant to boiling. So for example, water, even if you boil it, some endospores may still survive. The only way we can kill these endospores or get rid of them is to use an autoclave. and that's where we use pressurized steam. We heat it up to 120 degrees Celsius. It's under very high pressure, I think 20 pounds per square inch, and you leave it in there for anywhere from 20 to 30 minutes, and that will destroy the endospores. And again, you do that in something called an autoclave, and I'll show you the autoclave in lab if I haven't already done that. So I think I've gone through most of the structures that are listed on our bacteria prokaryotic cell up here. Let me look quick. So fimbriae, ribosome, cell wall, cell membrane, capsule. The cytoplasmic matrix, that is part of the cytoskeleton. We talked about the flagellum. We talked about inclusion bodies, the sex pilus, the chromosome. Actin filaments, these are also part of your cytoskeleton. They're just protein filaments that help organize the internal part of your cell. The mesosome, you don't have to worry about that one. And then we talked about the slime layer. So it looks like I've covered most or all of the different structures that you'll need to know. And again, these structures, you'll need to know the name of them, and then their structure, so kind of what they look like, but mostly their function, so what they actually do for that bacteria cell. So if you remember, in the first part of this chapter, we talked about cell wall, and one function of the cell wall is to help give that bacteria a shape. So up here we have common bacterial shapes, and most of our bacteria we're going to be looking at, they're either going to be the coccus bacteria or cocci, so they're circular or round, so they're sphere-shaped. We're going to have rod or bacillus bacteria. We're going to be looking at, these look like rectangles. The Vibrio bacteria, these are basically rod-shaped or bacillus-shaped, but they have a curve to them, very slight curve. And you can see that in the picture. We may look at some Spirillum bacteria. This is where the bacteria cells, they have a spiral shape to them. If they have extreme spiral shape, we call it a spirochete, which is in the middle bottom picture. So you can kind of tell spirochetes are very, very spiral, very compact. The Spirillum, they have just a few spirals to them. Some other bacteria, they're going to have branching filaments as well. We probably won't see any of those in lab. Mostly we're going to focus on if they're the coccus, if they're the rod or bacillus shaped, or if they're Spirillum shaped. So in lab, when you have to identify the different shapes, really focus on those three. Most bacteria, we can classify them into one of those 6 shapes that I just showed you. But other bacteria, they're pleomorphic, or they have a pleomorphism. This is when they have a variation in their cell shape and size in that one single species of bacteria. So this happens when you have bacteria that do not have a cell wall, and we looked at those in the first part of this chapter. So you're going to have these pleomorphic cells. They just kind of take on any shape. That's not any one shape. Besides having one of those standard shapes, bacteria cells also have something called an arrangement. And this is, so you look at the shape, so you focus on a cocci or coccus bacteria, so a circular bacteria cell. Those bacteria cells, they can just be in singles, so you can have one cocci, one coccus cell. If your bacteria cells, 2 of them hook together, it's called diplococci. So di is 2, and then cocci means that they're the round cells. So diplococci, you have a pair. If you have 4 of these cocci cells together, it's called a tetrad. So it's a group of 4. If you have irregular clusters, that is called a staff. So if your cells are clustering together, it's called staff. So staphylococci, those are regular clusters. If they're in a chain, it's called strep. So streptococci, your cells are lined up in a chain with each other. Sometimes if you have a group of 8 up to 64, they're called sarcina. It's a cubical packet of cells. They have like a cubical structure to them. And if you look on the picture on the right, there's the different examples. So up at the top, you can have one single bacteria cell. If you have 2 of them hooked together, it's called a Diplococcus cocci. If they're in a chain, it's called strep. So strep means in a chain. Part B, so in the middle we have our red cells. You can have your tetrad, that's when you have 4 hooked together. Or sarcina is when you have that cubical packet, and there's examples. And then our blue example, this is the irregular cluster, so you can have your staph. This is just when you have them all clustered together, and that's shown in the blue at the bottom. So we ran through the cocci, but the same thing with bacillus. So you can have diplobacillus, you can have 2 rod-shaped bacteria hooked together. If you have a chain of bacilli, it's called streptobacilli. So strep again is just a chain. If you have bacilli lined up on the long side next to each other, it's called a palisade. In order to classify all the different prokaryotic cells, we can use a lot of different systems or a lot of different types of information. So you can use their microscopic morphology. This is kind of what I just went through. So it's looking at the bacteria cell shape. So if it's the cocci shape, the bacillus, the spirillum shape, if it's a diploid, so diplococcus, if it's like streptobacillus, so you can use that microscopic morphology at the cellular level. The macroscopic morphology, this is when we look at colony appearance. So different colonies of bacteria, they can have different color, they can have different shape, they can have different smells to them. So you can also use that to kind of classify our bacteria. We can use bacterial physiology, so looking at if it has flagella, if it can produce an endospore. We can look at serological analysis and genetic or molecular analysis. So looking more at what types of genes that bacteria cell has, what types of molecules it produces, what type of different immunities it can have. One of the most common ways that we look at bacterial taxonomy is based on something called Berge's Manual. And it's called Berge's Manual of Determinative Bacteriology. It's a 5-volume book, and it covers all of the known prokaryotic cells. And I do have a copy of this manual in lab, and you'll be using it to get information when you do your Bacteria Unknown project the second half of the semester. So if you think about it, you can kind of look through it in lab for that. So in this book, the classifications based on genetic information or phylogeny. So they actually looked at the genes in these different species of bacteria, and then if the genes are more similar, they go into a closer group or closer classification. There's 2 domains in this book. There's the archaea and the bacteria. So far we've been talking about bacteria a lot, and that's what you're going to be working with in lab. In the next part of this chapter, we're going to look at the archaea so you can kind of understand what types of organisms those are. And there's 5 major subgroups and 25 different phyla under these 2 domains. And I'm not going to go into the specifics of it, but you'll see it when you use this book for your unknown project. So your major taxonomic groups in this, so you have the domain archaea. These are very primitive organisms, are adapted to very extreme habitats. They have extreme modes of nutrition. And I'll go through those in part 3 of this chapter. The domain bacteria, the 3 phylum, the 3 big phylum, we have proteobacteria. These have gram-negative cell walls. So all the gram-negative bacteria are in this phylum. The other 2 phylum, so phylum Firmicutes, they're gram-positive bacteria, and in their DNA, they have low guanine and cytosine content. So most of their DNA consists of adenines and thymines. The other phylum, the actinobacteria, they're also gram-positive, and they're the bacteria that have lots of guanine and cytosine in their DNA, and they have lower amounts of adenine and thymine. So, and they know this information because they've actually sequenced the DNA, and they can classify them into the two different groups, or the two different phylum up here. In the medical field, you're not really going to use Berge's manual. You're going to use more diagnostic scheme for medical use. So usually this uses more of the phenotypic qualities of the bacteria. So if they're gram-positive, if they're gram-negative, if they have flagella. And this scheme, it's restricted to bacteria that cause disease. So there are a lot of bacteria that don't cause disease that are included in Berge's manual. But for medical use, since you only are interested in bacteria that actually cause disease, you're only going to include those bacteria in this scheme. So this divides bacteria based on their cell wall structure. So again, this is the gram-positive, gram-negative. That's why that gram stain is so important. And then you're going to look at the shape of the bacteria. So if it's the cocci, bacillus, spirillum, the arrangement, so if it's like a diplococcus, streptococcus, staphylococcus, and then also other physiological traits. And this is probably really hard to see, but in your book in chapter 4, if you look at table 4.4, it shows different medically important bacteria. And first, they're based on their gram stain reaction. So the first group, they have a gram-positive cell wall, and then they look at arrangement of those cells. The second group has a gram-negative cell wall, so they're going to be pink after the gram stain. So you have the cocci, gram-negative, and then you have the bacilli or rod, gram-negative, and you have the spirillum. And then at the very bottom, you have your third group. These are the bacteria with no cell walls or special types of cell walls. And those are the mycoplasmas that we talked about in the first part of the chapter. Most of the time I'm going to be talking about different types of bacteria species. So just to define this, a species, it's a collection of bacteria cells. They share an overall similar pattern of traits in contrast to other bacteria whose patterns differ significantly. So a species example, I'm going to go with E. coli. Within a species, you can have a strain or variety, or you can also have a type within the species. So a strain or variety is just when you have a specific culture that's derived from one single parent cell, and that parent cell somehow differs in structure or metabolism from other cultures of that same species. So this is just when you have one bacteria cell that's a little bit different than all the other bacteria cells, but still has an overall similar pattern of traits. You can also have different types under a species. So this is a subspecies that can show differences in antigenetic makeup. So you can have different serotypes. They have different markers on their cell wall or on their cell membrane. They have different susceptibility to bacterial viruses or phages, or they differ in their pathogenicity, so how they actually cause disease, and those are different pathotypes. So in a species of E. coli, we have different types and strains of E. coli. So the strain or type of E. coli that can make humans sick is called E. coli H157. So there's different types or strains.